Hello everyone, this is Sheethal Maria with you at Bio Ocean, back with the continuation of cell series, and today we are here to learn about the residual part of plasma membrane, that is the molecular models of plasma membrane. In the previous video we just learned about the chemical composition, and now we are here to learn about the molecular models proposed by different scientists. And if any one of you are new to my channel, please go and subscribe to my channel and press the bell icon so that you can get the notifications whenever I upload a video. And before explaining about each models, I have to tell you that there are basically three models proposed by different scientists which are saying about the structure or the model of the plasma membrane, and here we are going to deal about those three models. Okay. And the first one among them is sandwich model, otherwise called as Danielli Davson model, which was proposed by Daniel and Davson in 1935. And this model states that the plasma membrane is a double layered structure, and the double layer is made up of proteins. In between the double layer of proteins there is a lipid layer, there is a lipid molecule layer. Okay, thus like it looks like a sandwich have two breads, and in between there is the filling, and the two breads is represented by those two protein layers, where the filling is represented by the lipids. Okay, and the inner end of the lipid molecules are non-polar, which are having hydrophobic tails, and the outer ends are polar with hydrophilic heads. Okay, these hydrophilic heads face outwards towards these protein molecules, so that these lipid molecules are linked to the protein molecules by these hydrophilic ends. Okay, the protein molecules and the lipid molecules are attached towards each other through these hydrophilic ends, and this arrangement of these proteins and lipids provides some pores in between, which will help in the passage of ions as well as water through, and together this constitute the sandwich or Daniel-Davson model. I will show you a picture so that you can understand it well. Okay, here I have shown the picture, and you can see that purple colored stuffs are those protein layers, and between there is a yellow colored substance, that is the lipid layer. And the lipid layer have a tail as well as a head, right? And those head molecules are polar and facing outwards towards the protein molecules, which are lying on the upper and the lower side of these lipid molecules. Now you are getting it, right? These lipid heads are facing towards the proteins. Lipid heads are in connection with the protein, that is shown in the purple layer. That is how the passage of these ions and water are progressing properly. Okay, this is the model. And then comes the second model, that is unit membrane model, otherwise called as Robertson model, which was proposed by Robertson in 1953. It states that plasma membrane consists of two layers of molecules, and the two layers of molecules are proteins, one on the inner side and other on the outer side, and a lipid bimolecular layer is there in the central region. As I have stated in the sandwich model, there are two protein layers, inner and outside of that, and in between there is a bimolecular layer of lipid. It forms the central region, and the protein layer have a thickness of 20 angstroms, while the lipid layer have a thickness of 35 angstrom. This is also called as a trilaminar model, that is having a three layer. So that is why it is called as trilaminar model. Otherwise we can call it as unit membrane model or Robertson model. Okay, hope you understood. For better understanding, I will show you a picture. See the picture here. Those orange colored ball-like substance with a tail, that are the lipids which form the central region, and above it and below it there is a green colored portion, that are two protein layers, same as that of sandwich model. Okay, upper and lower side are proteins, in between in the central portion there is the lipid layer. That is what we call as unit membrane model. Okay. And the third model is fluid MOSAIC model proposed by Singer and Nicolson in 1972. And this model states that plasma membranes are quasi-fluid structures in which proteins and lipids are organized in a mosaic manner. And this is the widely accepted, and we all much depend on this model to study about the plasma membrane. Okay, and we are going to study in detail what is that structure and all those stuff. It is much complicated. Okay, you have to keenly listen to what I am saying. Here in the fluid mosaic manner, there is a layer of lipid, and that lipids are phospholipids. They reside at the center, and surrounding these phospholipids there are protein molecules which are arranged at different ways at different regions of plasma membrane, while some protein molecules penetrate into these lipid molecules partially, otherwise fully. Some goes partially while some goes wholly. And these protein molecules which penetrate inside the lipids are intrinsic proteins, while those proteins which lie outside without penetration on the lipid bilayer are called as extrinsic proteins. These extrinsic proteins are actually globular or spherical in structure and bounded to the lipid layer very loosely, while those intrinsic proteins which are penetrated are tightly bounded to the lipid layer. The hydrophobic tails of those phospholipids face each other internally. Those tails are facing each other internally, while the hydrophilic heads faces the proteins which are residing on either side of this lipid membrane. Okay, and in between these phospholipids, molecules of cholesterol and glycolipids are also seen. These are seen only in animal cells. Okay, this is for an additional information for you. I will show you a picture for better understanding. Okay, here it shows a picture that is having blue colored structures with a head and a tail, that is the lipid layer, as well as there are so many proteins like peripheral protein, channel protein, carrier protein, like that so many proteins are there, and you can see it's on the either sides, as well as there are cholesterol like substances in between. I have shown those peripheral proteins here now. They are seen on the inner surface, while those integral proteins, those channel proteins and carrier proteins together called as integral proteins, they form a channel for the passage of ions. Okay, so on the peripheral protein on the outer side, there are so many carbohydrates attached to it. Proteins are attached to carbohydrates also in this model. Okay, this is what fluid mosaic model is. Hope you all understood about these three models. This is all for today. Let's see you in the next video. Thank you for listening. If you like this video, give me a thumbs up, comment your suggestions, and share it to your friends and others who are interested to learn biology. Let's see you in the next video with another topic. Thank you for listening. God bless. Thank you.